So in our previous unit or lesson, we talked about primary structure and secondary structure. So primary, just remember, it's just a string of amino acids. It's the sequence that you have of the 20 amino acids. Then you have secondary structures, which is when it starts folding upon itself. So the common ones is the alpha helix, which is pretty much kind of like a double helix, but it's just with proteins. You could also have beta strands. That's another structure that you see. And these are very common kind of pictures that you'll see when you look at protein structures, at least a big tertiary or quaternary one. Here's a kind of a super complex of secondary structures. So you can see here, you have a bunch of beta sheets right here. Here you have 4 alpha helixes, and you can actually have a combination of both. So these are ways that proteins can fold on themselves, and then the last one that I was going to show are the turns. So the turns are like prevalent here, they're turning here. So just areas of amino acids that allows the strand to kind of flip on itself. So here is an example of a turn. So specific things, and they'll have interactions that will kind of keep it together. Now, why is this important? Well, when we start looking at bigger pictures or tertiary structures, is instead of just having these secondary structures of alpha helixes or beta strings, now you have these really complex, as the amino acid gets longer, these complex structures that could do things for us. So it's a combination of alpha helixes, beta strands, turns, and just things that help it do the enzymes or protein folding that it needs to do. So protein folding is actually pretty complex. You know, you're starting from a very long unfolded chain of amino acids into this, and sometimes they could be misfolded, sometimes they will be folded with help. So you have things like chaperones or chaperones that are proteins that help fold more proteins. And also when you're making proteins and during the translation process, it's coming out one at a time, so it's not the whole string of protein that's available. So here, you know, the whole protein is available and it's going to start folding on itself. Well, what happens if it does one interaction that it might have not if it was being produced? So remember, when amino acids are being produced, it's starting from one side and slowly growing to the other side. So it's not the whole thing is present and then it's going to fold. It's going to start folding the moment that one piece is made. So let's say the reddest piece is the part that's ready. So first it'll be made. That part could now be folded on itself, so it's going to get bigger and bigger. So a lot of times, even when we watch the video of the egg being folded and unfolded, sometimes it can't be folded back because you have the whole string of amino acids versus just part of it. So it just depends on the protein. It depends what's there. So a lot of times you're going to need some help to kind of fold the protein itself. So you have things that help fold it correctly. You also have things that try to get rid of them. So proteasomes are another type of amino acids that go after, that break down amino acid chains that are improperly folded, or proteins that are just old and not useful anymore. So they'll be targeted. So proteins that you don't want anymore, your body will target them, and then these guys will come around and degrade them. So that's really cool that you have things like that part of it. You could have a protein and you, with certain kind of chemical reactions, heats, you could unfold it. So the video that we're going to watch with the eggs, that's exactly what's going on. You have a protein that's cooked. So when you cook eggs, it becomes cooked, and then you can uncook it by doing certain things. So here's our protein. Heating it will change it. And then for eggs, the protein will change shapes. When you cook protein like chicken, it changes color. You can even have those juices, right? And then when you boil chicken, you kind of have those solid things that kind of float out, and that's just because in the enzymes or in the liquid, the protein didn't seize up yet, but once you cook it, it does. So here's an example of a protein with many different structures. So here you have hydrogen bonds on this side. Here you have a disulfide bond, which we talked about in hair. This is actually really strong. But the rest of these aren't that bad. So metallic bonds are kind of more ions. Ionic bonds are kind of important. But these are easily broken up by heat. So that's what's happening when you cook something. Other things, you need some chemicals. So disulfide bond, remember we needed lye, or some really strong base. Ionic bonds, usually if you put it in the buffer or like an ionic solution, will help break it apart. Sometimes you need certain things for ionic bonds. And even soaps. So soaps could break down things. Parts of the hydrophobic places can now be exposed. Things like that could happen. So here's kind of a list of kind of different things. So you can have the first two, which are reagents that could break down disulfide bonds, but these are things that we might use in a gel. So second semester we'll probably have, if we're back into the classroom, we might do electrophoresis, a gel, where we separate either DNA or protein, and we're going to have this to kind of break apart our proteins so they're a nice long strand versus all clumped together. Detergents like soap help break apart things. So normally when proteins are in water, they're going to fold a certain way, but when you put them in soap, they're going to unfold or fold in other ways, because now the water parts or the hydrophilic parts are going to try to get away from the hydrophobic stuff. So that's really cool in itself, and that's why you use soaps to wash your hands and stuff. Not only does it help loosen up dirt from your skin, but also it can have a possibility of denaturing proteins. Same thing with alcohol. Alcohol can denature proteins or in DNA. Most likely it's proteins, and that helps disrupt or kill the bacteria or viruses that are there. Heat is another way. For most parts, low heat is necessary for hydrogen bonds, but as you want to break chemical bonds, then you require more and more changes. Now, when it comes to just heating it up, if you really want to break the chemical bonds, you really need to heat it up and cook it. pH is another way of changing it. So in the egg cooking video, you kind of see you could cook foods by pH. Ceviche is one example. You put things in acid and it could cook your food that way, because the acid will change the way the proteins are, and you could break it down that way as well. So you have different ways of breaking down proteins. So it's pretty interesting what you could use. Here are kind of some examples. Now, the really scary part is these are if proteins are folded correctly, and then ways to break it down. What happens if a protein is folded incorrectly? So prions, kind of what made mad cow disease, is a protein that's folded incorrectly. If you just have one, it's not a big deal. That protein's not functioning correctly, so either an enzyme is not working, a reaction is not working, or parts of a cell is not going to function correctly. But if it's just one protein, even a cell will be fine. It's just one. But what happens if this protein can make other proteins misfold? So this protein, by touching a normal protein, can have it misfold into a bad one. And that's what mad cow disease is. You have a proper protein, and then when it comes in contact with misproper, the mad cow protein, then it's going to all become that. And that's the danger of it. So here is how it should look like. Gets infected, and then what's going to happen is it's going to change all the proteins into a useless or degraded protein or prion. So that's kind of scary. We also are finding that a lot of our brain diseases are caused by misfolding proteins in our brains. It might not be as crazy as prions, but there is significant changes in our proteins. So scientists right now, researchers are trying to figure out how to undo it or stop these from forming more and more. So as these proteins are kind of blocking your normal brain function, then you kind of have these brain diseases where it's really sad, where the brain just starts degrading over time. We're going to explore that with Kuru and also with CJD. These are kind of brain diseases that are more similar to prions. So it's going to be really neat to kind of look into these things. And to me, this is really cool of how when proteins are folded correctly, they work wonderfully. But when they don't, it could do just as much damage the other way around. So hopefully you enjoy the videos that I have planned for you.